A Carnegie Institution for Science audio press release for June 5, 2014. MAD. Scientists shed light on braking mechanisms in cellular signaling. A team of researchers studying a flowering plant has zeroed in on the way cells manage external signals about prevailing conditions, a capability that's essential for cells to survive in a fluctuating environment. Researchers at the University of California, Berkeley, the Plant Gene Expression Center, University of California, San Francisco, and the Carnegie Institution for Science identified a novel mechanism by which the strength of such an external signal is reduced or attenuated. Their work focuses on the tiny mustard plant Arabidopsis, which is frequently used by scientists as an experimental model. Their findings were published in the journal Science on June 6th. Attenuation of signaling is analogous to the brakes on a car. While acceleration is desirable, acceleration without restraint can be disastrous. In this research, Arabidopsis seedlings were taken from subterranean darkness into sunlight, which triggered a response leading to rapid and extensive redirection of gene expression, ultimately resulting in familiar green seedlings. But a break on this acceleration of new gene expression is also necessary to restabilize the cells at a new equilibrium. The research team discovered a nuclear-localized bimolecular signaling configuration by which the braking mechanism is directly linked to the accelerator, thereby providing simultaneous acceleration and restraint. By identifying the mechanism involved in this attenuation process, the team's discovery has potential implications ranging from agriculture to cancer research. Cellular signaling triggered by external cues such as sunlight enables organisms to adapt to prevailing conditions. When the organism perceives something that requires a response, a series of chemical signals is activated. This signaling is generally very robust at first, but at some point it's necessary to dial it back or turn it off entirely, a restraint that falls to different, less understood signaling pathways. These types of restraint functions are of great importance but poorly understood, as scientists have focused mostly on how the cells get stimulated in the first place. Light signaling in Arabidopsis involves the binding of an activated photoreceptor molecule called phytochrome to a transcription factor or gene switch called PIF. This binding destroys PIF, switching off its target genes. However, the researchers found that in imposing PIF's destruction, phytochrome signed its own death warrant and is simultaneously executed, thus reducing the incoming light signaling intensity. Carnegie's Zhang Wang, one of the study's co-authors, notes that the understanding of such molecular mechanisms underlying light response kinetics is important for engineering crops that can better adapt to environmental fluctuations. This bimolecular mutually assured destruction, or MAD, mechanism of signaling attenuation, appears to represent a new configuration, thus broadening our understanding of the range of mechanisms nature has evolved for this critical function. With the independence for nimble pursuit, Carnegie scientists tackle some of the most profound challenges in modern science, fundamentally changing what's possible for us, our planet, and our universe.